All right, the third video I want to talk about, finish up this idea of transcription, which is basically making RNA. The genes, the information to make a particular protein are in DNA. DNA is going to stay in the nucleus of the cell. The cell wants to keep its DNA protected and in the nucleus and use something else to take that message out to the ribosomes, which is where these proteins are being synthesized. So that's what we're going to look at. That's what we're going to see is making RNA, and then that RNA is going to go off out to a ribosome, and in translation, the next phase that we're going to talk about on Friday, then we'll see what that RNA does and how that information is conveyed from RNA to a chain of amino acids in a protein. So first of all, this is what happens at the site of DNA. You've got DNA, this enzyme comes along and splits DNA apart. It breaks the bonds between these bases. And now those bases are sitting out there flapping in the breeze. You've got a base in DNA that's an A. It wants to form a bond with a T if it was looking for more DNA. But in this case, there's an enzyme that's coming along that's bringing one ribonucleotide in which there's no T's. There aren't any T's. There's U's instead. So this enzyme is going to bring a ribonucleotide, and by that I mean a sugar and a phosphate and a base. And the base is going to match up. It's going to pair with the complementary base that's in DNA. So you've got one base in DNA and you've got the other base in RNA. They match up with one another. It's not just any base. You have the same pairing idea that one base is only going to form a bond with the one other base. Now, there's 4 different types of bases, and one base is specific for one other base. The other 3, it's going to ignore. It's not going to form a bond with. So that's what happens here. This is a strand of RNA that's being synthesized. One little ribonucleotide comes in and forms a bond, this pink one with the green one. Earlier, this blue ribonucleotide, whatever base that is, matched up with this red base. Here's a blue one with the green one with the blue one. So the bases that are matching up here, this blue base is only in DNA is going to match up with a very particular base in RNA. These are coming in one at a time, one nucleotide, another ribonucleotide because it's RNA. And then these are being joined together to form this temporary strand of RNA. When this is done, then this messenger RNA is going to leave and the DNA is going to go back to its original form. So let's look at this in a little more detail then. This is what I was talking about here, that here's this strand of DNA. In between this green rectangle and this red rectangle is the information that DNA contains to make a protein. So in other words, that's a gene. That's a segment of DNA that can be used to make a protein, among other things. So here comes this RNA polymerase. This DNA molecule is being split apart. These bases in the DNA are sticking out there, looking for a pair. And this is something similar to what we saw in DNA replication, but in this case, this is transcription. This is making RNA instead of DNA. So here come these little ribonucleotides. These little T-shaped things have a sugar and a phosphate and a base. The base is either an A, or it could be a U, or it could be a C, or it could be a G. But these bases come in and they pair up with these exposed DNA bases very specifically. And so you've got this messenger RNA molecule that's being synthesized all along here. These little ribonucleotides are coming in one at a time and being joined together to form a chain of ribonucleotides. And this enzyme is going to move along the DNA molecule all the way to the end, and it's going to get to this point where there's no match, and this is going to be a signal for proteins or the synthesis of messenger RNA to stop. This is bringing in one ribonucleotide, another one. It's going to proceed all along here until you get to this termination site where there's a signal for this whole process to stop. So let's look at that in a little more detail. Here's the strand of DNA. This RNA polymerase would have moved along this strand of DNA, breaking the bonds between the bases and allowing one ribonucleotide at a time, one sugar and a phosphate and base, another sugar, phosphate and base, and then they're being joined together to make this chain of messenger RNA. These are ribonucleotides. This is deoxyribonucleotides. This is a chain of nucleotides stuck together, and so is this. It's just a different type of nucleotide, ribonucleotides instead of deoxyribonucleotides. So now this enzyme comes off, DNA, the bonds between the bases form again, and DNA goes back to storing the information that it's done ever since the beginning of the cell, ever since the beginning of the organism. This messenger RNA is done. It's been made, and transcription is complete. So now this messenger RNA is going to leave the nucleus and go off away from the DNA, and it's going to go out to the ribosome outside of the nucleus of the cell. What it has in it are the instructions for making a protein. These sequence of these bases, this blue and red and yellow and orange thing that are sticking up, those are bases. They're not just any order. They're in a very specific order that's been determined by the order of bases in DNA. They are different bases, and these are different ribonucleotides, but the information is the same. And from the beginning to the end of this messenger RNA, that's information that was in a gene. And from the beginning to the end of this messenger RNA, that's information that's going to be used to assemble a chain of amino acids and make a protein at the ribosomes. That is what happens in translation. This is transcription, where the language is nucleotides in DNA and the language in RNA is nucleotides. They're different types of nucleotides, so it's just transcribing rather than two completely different languages like nucleotides and amino acids. So transcription is done. What you've accomplished as a cell is you've made messenger RNA. Not just any messenger RNA, but a very specific strand of messenger RNA that contains information in a gene, basically. Now the information in DNA stays in the nucleus of the cell. It's confined there. It goes back to being DNA, the storage form. It's not going to go off and be altered or be messed with at the site of ribosomes. So now the next step, if you're a cell and you're going to make proteins based on information in DNA, is to take that information in RNA, deliver it to ribosomes, and make a protein there. So that's what the next step is. And talking about the languages again, DNA is a language. It's a repeating subunits of nucleotides, deoxyribonucleotides. RNA, repeating subunits. The language is nucleotides, ribonucleotides rather than deoxyribonucleotides, but nucleotides nevertheless. For proteins, we're talking about a completely different language with a completely different number of letters, 20 letters for the 20 naturally occurring amino acids, as opposed to 4 letters, which are the bases in the nucleotides in RNA, and there's 4 letters, 4 bases in the nucleotides of DNA. So this has to be translated, that's why this is called translation, from one language to the next, from a language of nucleotides to a language of amino acids. So this is what we're going to look at next, is making proteins. So far, we looked at the function of RNA and saw how RNA is necessary to make proteins. DNA is just sitting there in the nucleus of the cell, storing all this information. You need to get that information from the DNA out to the ribosomes. That's what RNA does. We looked at the structure of RNA and saw that it's got some differences between DNA. It's a single strand. It's got a different base. It's got a different sugar. The lifetime of some of these RNAs is short compared to DNA. And there's 3 different types of RNA. Those are going to be important. These 3 different types are going to be important when we look at making proteins, because all 3 of those, messenger RNA, mRNA, that's the same thing, messenger is M, transfer RNA, which is abbreviated as tRNA, and then ribosomal RNA, those are all 3 involved in making proteins in the ribosomes. So this is where we are so far. We've gone from DNA to RNA and transcription. Now we're going to go from RNA to making proteins and translation. The first step of expressing a gene, so taking the information in DNA and making a protein, the first step is transcription. That's where we are right now. We made this, you've kind of copied the blueprint that's in DNA to some form that you can leave the nucleus with, which is RNA. Now, the next step, as I said, is to use all 3 of these different RNAs, especially the message that's in messenger RNA, at the site of the ribosomes and make a protein. So that's what the next lecture is going to be on, is that whole process of translation. Transcription and translation, they're both phases of gene expression or protein synthesis. It's kind of like mitosis and meiosis for some people in this class. They know they start with a T, it's trans something, ends with an N, and they don't know what the difference is. But if you look at these, if you map these out yourself, if you make sense to yourself what's happening in transcription and the 3 different types of RNA that are involved in translation and how that works, getting to a chain of amino acids, then you'll have a good understanding of all of this. So that's what we have to look forward to on Friday.